So now that we've set the stage with all of the anatomy, we can actually talk about the digestion and absorption that happens in your digestive tract. So here we're going to start by talking about macronutrients. So when we say macronutrients, we mean nutrients that are big, that you need large amounts of, and most importantly, that provide calories. And those are carbohydrates, proteins, and fats. Now this should sound familiar because we're coming back to what we talked about with that digestive enzyme lab. So we're back looking at this whole tube, and we're going to talk a little bit about where in the tube breakdown and absorption of each of these happens. Now we're going to start with an important equation that applies to everything we're looking at, and this is hydrolysis. So hydrolysis is when we take a large macromolecule and use water and a digestive enzyme to break it into two smaller molecules, one of which has an OH group and one of which has an H. And so that's hydrolysis. Basically, we've inserted a water and used that to break it apart. So here we're talking about like a protein, we're breaking into smaller peptide. A carbohydrate, we're breaking into smaller carbohydrates. A fat, we're breaking into smaller fats. And that's what we're going to be seeing with this whole process. And the other important thing to talk about is absorption. We're going to be talking about digestion and absorption together because the final stages of digestion lead directly into absorption. So for something to be absorbed into the body, it has to come out of the area where all of that food in the process of being digested is and go into our bloodstream or our lymph to be carried off to other parts of the body where it's needed. So inside these intestinal villi, which we saw last time in the small intestine, there is a dense network of capillaries that goes right up against the surface where stuff can be absorbed in. And we also have the central lacteal that goes into the lymphatic system. And fat is all going to go into there so that it's not clogging up our bloodstream. So we're back in this, inside the villi, we have this lacteal, we have these capillaries, and then on the very edge we have this brush border. And that, again, maximizes surface area. But you're also going to need to remember that brush border because we're going to come back to it when we start talking about some of those final digestive steps, because some of those final digestion steps happen at that brush border along with absorption. All right, so we're going to start with carbohydrate digestion because it's kind of the simplest. Carbohydrate digestion, we start with these polymers. Polymers are lots of single units, lots of monomers linked together in long chains. Now, in this case, some of our most common ones are starch and glycogen, which are polymers of simpler sugars. And those simpler sugars particularly are disaccharides. So disaccharides are two monomers, two monosaccharides linked together. And we're looking here at maltose, sucrose, and lactose. All of the simpler sugars that we eat are disaccharides, like your table sugar is mostly sucrose. And then these break down into monosaccharides, glucose, fructose, and galactose. Now, lactose you've almost certainly heard of before because lactose is one that a lot of people have a problem with if they don't have lactase, this enzyme that breaks it down. So you'll see we've got these digestive enzymes here. So here's amylase. Amylase breaks down starch into disaccharides. And amylase starts in your saliva. You've got salivary amylase. If you ever take like a saltine, a very simple white flour cracker, and let it sit in your mouth for several minutes, it's kind of unpleasant, but you'll start to taste sweet. And that's because that amylase is breaking that down into these disaccharides. And then we've got specific enzymes that break the disaccharides down into monosaccharides that we can actually use for energy. Now, to absorb those monosaccharides, we need transporters to get them into the cell. And we have a transporter that works on fructose on one side and glucose, galactose on the other, and a separate transporter that is our what we call a sodium glucose symporter. So all of these are active transport. So we've got active transport pulling this in. There's different ways of doing this. One is by using ATP to actively transport it, and the other is by using ATP to generate a gradient and then letting something fall down that gradient, pulling something up its gradient with it. So in this case, we're creating a sodium gradient where the sodium level is lower inside the cell using this sodium-potassium symport that you've seen before, the sodium-potassium transporter, sodium-potassium pump. And so that pumps sodium out, potassium in, and then we can let sodium fall down its concentration gradient in, and then that pulls glucose and galactose actively up the transportation gradient. And fructose, we basically just sort of let fall down its concentration gradient, but we diffuse it into the blood very quickly so it doesn't build up in the cell. Glucose is sort of our most important thing, so we're really focused on getting that in. Now, the other important piece here is the brush border enzymes. Remember I said that brush border was going to come back? So here's where that brush border comes back. And the brush border enzymes are actually where the very final digestion step happens. So these are actually in the membrane, and they're going to do this final breakdown of these disaccharides into monosaccharides, which they're then going to send directly into nearby transporters. So this final set of steps happens right in the end, in the brush border. And this lactase enzyme in the brush border is the one that most people don't produce as adults. Everyone produces it as babies and young children, but most adults don't actually produce lactase. It is lactase persistence is something that some people, groups of people have evolved to allow adults to continue producing lactase, to allow us to eat dairy products and get all the nutrition from them. So those are our brush border enzymes, and that's our carbohydrate digestion. Now we can move on to protein digestion. Now proteins have an added complication. They're still these long, complicated peptides just like starch, but they also have extra structures, and those extra structures are protective. All of those folds and bends, and all those things that are really important for making an enzyme take the right shape and stuff, that makes it harder to break down. And the other complication is we don't want too many enzymes that break them down, because if you have enzymes that break down proteins, they're also going to break down your own proteins. So we have to be really, really careful about this. So what we do is we have only one enzyme, pepsin, that can break down whole complicated structured proteins into simpler polypeptides. And that enzyme is only active in low pH in the stomach. And so it can't break down proteins anywhere else. It can't break down the rest of your body. And it breaks these into polypeptides that then trypsin can break down. Trypsin can't break down these complicated structures, but it can break down polypeptides into smaller peptides. So trypsin is secreted by the pancreas, it's active in the duodenum, breaks down the polypeptides into smaller peptides. Then in the small intestine, we have orepsin, which breaks down these smaller peptides into individual amino acids. And that is what we can finally absorb. So here's our structure. We have these proteins, they come in, they're not affected at all until they get to the stomach. In the stomach, they're denatured, so they're taken, the shape goes away, they're partially hydrolyzed, which means we start breaking them up. And then, so remember that hydrolysis reaction, right? So hydrolyzed, hydrolysis. And then the small intestine, we have trypsin and chymotrypsin and aminopeptidase and carboxypeptidase, and all of those start breaking these down into small peptides and amino acids, and they're actively transported into the bloodstream. So looking at some of these pieces, in the stomach, we have pepsin, which is produced by the chief cells as pepsinogen, and then activated by the HCl that's produced in these parietal cells. We're back in these gastric pits. And this is only active with this high levels of HCl. And of course, this is this enzyme optimal pH that you've seen before. So we can see that pepsin is only active in this very low pH. Amylase, the starch enzyme, is somewhat intermediate, and then trypsin is active in a higher pH range, because at this point we're in the small intestine, and the pancreas has actually secreted bicarbonate to bring the pH up so high that pepsin has no activity at all. So you see, once it gets above 6, there's no activity at all. Now the pH in your small intestine is usually around 8 or above, so trypsin is maximally active, pepsin is not active at all. And then we come back again to brush border enzymes. So just like with the carbohydrates, our final step in the protein digestion actually takes place in this brush border with these aminopeptidases that are going to break down small peptides into individual amino acids. And then those individual amino acids can be actively transported into the cells. So here we're seeing another sodium symport. So just the same as with the glucose, we're pumping sodium out, then we're letting it fall down its concentration gradient, carrying amino acids up their concentration gradient into the cell, and then they can fall down their concentration gradient back into the blood. So that's protein digestion. Then finally we move on to fat digestion. So fat digestion, you have to remember, why is this particularly difficult? Now the problem with fats is that they're not water soluble. So if you've got oil and water and you mix them up, the oil is going to reform these large droplets. Now that's a problem because our enzymes are water soluble and they can only get to this very outer layer of this large fat droplet. So we need to break it up, and for that we use these bile acids, these bile salts, coming from the liver stored in the gallbladder, to break these up into smaller particles that then the lipase from the pancreas can act on and break the fats into monoglycerides and fatty acids. And then those are absorbed through the villi via micelles, which we'll talk about that in a moment, and reform into triglycerides. So those are the types of fat that we actually carry and use for most things. And we've got cholesterols here too. These aggregate, they're combined with cholesterols, proteins, and phospholipids to form chylomicrons, which are going to be carried around. So now what are we talking about with these micelles and chylomicrons? So both of these, we're looking at these sorts of phospholipid structures. So we have phospholipids on the outside, where we've got the hydrophilic heads on the outside and the hydrophobic tails on the inside, creating the structure where fat can be carried around on the inside. And all of that allows us to carry fat around in the lymph and in the bloodstream, and use that fat without it forming these big clumps of fat, because if you have a clump of fat in your bloodstream, that's going to come out of solution, and then that can become a blockage. That's why cholesterol can be a blockage, and saturated fats, which are more solid, can be a blockage and cause you problems.